Hello, everyone. Thank you for joining and returning. So this really shows how the community is supporting the other ones that haven't presented yet. So let's wait a little bit to see if some other ones are joining. And as you saw during the last 2 days, we have these 10-minute presentations, but it's hardly enough time to continue discussing and everything. So I guess we will just start with everything. So first of all, welcome back everyone to this virtual ECR meeting. And this is the third day. And we're really happy that it's the last one and the final. So we have had really great presentations. Thank you all for this hard effort. And for the ones that are going to France, we are very confident that you're going to present exactly the same. A little bit more nervous maybe, but we're going to be there and to support you. And yes, so we are looking forward to that also. So today's program, don't forget about the consent on your YouTube presentations. Paula is going to send an email later on or tomorrow, sending you like the latest news and the certifications of assistance and different informations. For example, that the YouTube presentations are going to be 2 weeks. So that everyone can see them and continue, commenting on them. So we're starting right away with the second part of the session on paleolimnology. And then we will follow with the keynote, with the first one of Camila on tectonic and climate signals during the Miocene in Patagonia. Then we will continue with the part 3 of the same session, paleolimnology. We will make our group picture and a 15-minute break. We will return with Xavi's keynote on open paleoecological databases. Finalize then later with the session of paleolimnology. And then we have one last session that are on related topics, like some studies that are modern, but everything it's really needed also to improve the interpretations on our paleoecological records. And then at the end, Cecilia is going to give some final words and also talk a little bit about the Limnogeology Association. And we will wrap up everything and say goodbye. And so with this, we go now to the 10-minute ECR presentations. Remember the rules, make your questions at the end, unmuting yourself or via chat. And so now I'm asking Angie if you can chair this session, please, together with your colleagues.
Yes. Hello, everyone. Welcome to the session 2, Paleolimnology, Reconstructing Environmental and Climate Change. Today we are going to continue, as tomorrow, exploring how lake sediments serve as natural archives, preserving evidence of past environmental and climatic changes. For today, the first presentation is from Juliana Velandia and is titled Mid to Late Holocene Climate of the Northern Great Plains, Southern Saskatchewan, inferred from multiproxies in lacustrine sedimentary records. Juliana, are you there?
Yes, I'm here. Can you hear me well?
Can you share your screen, please?
Yes.
And good luck to everyone.
And, sure. Can you see it now? Is it in the presentation mode?
Yes.
Okay.
Yes, perfect.
Good. Okay, so thanks for being here. Today I'm going to talk about my master project. So I'm Juliana Velandia and I'm a PhD student at Memorial University. So my project for my master was to meet the mid to late Holocene climate of the Northern Great Plains in southern Saskatchewan, inferred from multiproxies in lacustrine sedimentary records. And I want to acknowledge that this research was carried out at the Treaty 4 and Treaty 6 territories, the homelands of the Nahiwak, Anishinaabe, Dakota, Lakota, Nakota peoples, and Michi Métis nations. And I want to acknowledge all the institutions and organizations that were involved during this project. So first of all, the study site, it is in the northern part of the Great Plains in North America, and it's southern part of Saskatchewan. I have one lake, Deep Lake, and it's not very deep. It's only like 2.5 meters, and it's towards the east of Regina. And then on the left, we can see Saint Denis is a pond that belongs to National Reserve area towards the east of Saskatoon. So the methodology that I used was first the core extraction as an archive, radiocarbon dating, XRF on the sediment, and then my main tool was ostracods. So I use the ostracods identification, and based on the most abundant species, I perform stable isotopes and trace elements via IAL-IPA in the carapace, and then these in order to have the paleoenvironmental reconstruction. So the radiocarbon dating show us that Deep Lake in these around 195 centimeters, we have 6,700 calibrated years before present. Two hiatus were inferred here. And for Saint Denis, this core has around 1,017 calibrated years before present. I performed some PCI analysis of the XRF, and I found some elements associated with chemical precipitation, runoff, and interestingly, I have for Saint Denis, I have the silica has an interesting behavior. So infer these so far with the information we have as a biogenic silica, but also we have here the zirconium, which we were not expecting to be in the chemical precipitation. It should be on the runoff and catchment erosion, but this is something that I am working on needs further research. So for the ostracods, I use a resolution every 5 centimeters, and I identify based on the abundance of the ostracods two main zones for Deep Lake. The most abundant species is the Fabaeformis condonobtusa, and we can see it here on the right, a male on the top and a female on the bottom. Then for Saint Denis, the most abundant species was Cypridopsis virua, also with Condona acutula, and I identified 3 main zones. The analysis were performed in Cypridopsis virua. So for stable isotopes, I found some trends. So the stable isotopes for Deep Lake were only available until 150 centimeters due to the abundance of the ostracods. And we can see here some trends in the carbon and oxygen, mostly more detail for carbon, because the ostracod oxygen stable isotopes are showing more variation in Deep Lake. But we can see there is a trend in Saint Denis for both carbon and oxygen in terms of evaporation and precipitation, and more DIC content, more or less DIC content. So I wanted to show what I did for the methodology for the trace elements in the carapace, because I find it kind of cool. So I did it through laser ablation, ICP-MS, and this is the way in which we assembled the ostracods in a double-sided tape. I took after that the micro XRF mapping to see which valves were destroyed after the laser, and those that were destroyed, I disregard those valves for the analysis. And I also, before that, I had a map of which valves were from each stratigraphic layer. Based on the trace elements, I use magnesium, barium, strontium for salinity, and manganese, iron, and uranium for redox conditions. Again, we can see here some trends and some two periods, important periods associated with salinity. These are strontium. So strontium, because it can be influenced, but when there is aragonite precipitation, as well as barium. So I didn't use these two elements too much because it could be the information about salinity could be biased. But I have here some redox conditions, and likewise for Saint-Denis, I also have two periods in which I can see changes here. So then I put all of the proxies together for the bulk sediment chemistry that I found it the one that I didn't want to base all my interpretations. Very important because of the resolution that I had. So my main proxy were the trace elements, and then I compared that with the information that I have for the other. So I can see here some important periods, and then likewise for Saint-Denis. In Saint-Denis, probably recording the medieval warm period in which there is a higher salinity, and this is also supported by higher operation according to the oxygen. And for the Little Ice Age, there is, yes, a higher salinity, but the stable isotopes are not really showing, at least not at this resolution, many or very detailed changes. So I put everything together, and just to picture these changes in southern Saskatchewan, I found more some centennial cyclicities changes in which I have humid periods and more centennial cyclicity changes between dry and humid. Dry here, and then towards the Little Ice Age associated with weak westerly winds according to previous works. And then more cyclicity again during the medieval warm period when there is a record of more stronger westerly winds. And the two hiatus that I forgot to mention before, so they're here, and here. Even though I just have these few radiocarbon dates to infer these, I compare these with other works and research that have been done in the Northern Great Plains, and I found that this coincide with some other works. So just as a conclusion, the mid-Holocene was mainly arid punctuated by centennial wet periods, and the late Holocene displayed seasonal changes between dry and wet periods. But just to take into account, this is just for two little lakes. I did compare these with some other works, but this is probably these are responding to more local than regional changes. And one of the more relatively novel technique will be the LAICPMS in the carapace. And I found the most reliable of magnesium calcium for salinity. And some future research, I'm still working on these in order to be able to have a better reconstruction of the Northern Great Plains. And thank you very much for listening. So happy to take any questions you have.
Thank you, Juliana. Everyone have questions?
So I have a question. Thank you, Juliana, and really nice presentation. Which one is the resolution of the XRF analysis that you carry on?
It was also every 5 centimeters.
The same resolution as the obstacles. Okay, cool.
Yeah.
I have a question. Oh, no, sorry.
Yeah.
Go.
No, go ahead, Marta.
No, I just want to ask if there are any specific effects when you analyze magnesium, calcium, or trace elements.
Mm,
If you always use
what?
the same species and you have to correct the data, like for example, in stable isotope, you have a vital effect and you have
Yeah.
to correct depending on the species. If you, this is the same for trace element.
That's a good question. I didn't find that in the literature. I didn't find that that was the case. Um, like there was no previous correction with the viral effects. But this is something that I should look into, like for the future. But no, I didn't find anything. I will think, or from what I known, the stable isotopes due to these fractionation that is in equilibrium, they can be affected by viral effects. But I don't know if that's the same case for trace elements, as they are more accurate when they are being incorporated by the carapace.
Maybe you can do the test using different
Yeah.
species and see if you see any differences.
Yeah, that's a good idea.
Thank you.
Okay.
Okay. I was just wondering if you have considered to perform clump isotopes in the ostracoval bulbs.
Yeah, I did. I was very ambitious when I had the idea. I know about Marquillano work, and I was very ambitious to follow her work. But then I actually met with her. We talk about it, and it would be worth it, as it hasn't been done, or at least not to my knowledge, it hasn't been done in this part of southern Saskatchewan. But probably due to the resolution that I have, it wouldn't be very worth it. And also because the material, like we need a lot of valves. So yeah, but it will be interesting if I had like a longer core.
We can discuss if you want.
Yeah. I still have the ostracods like a lot, and I don't know what to do with them, so I would love to.
Okay. Well, thank you, Juliana.
Well, thank you. I'll stop sharing.
Okay.